good to see you. Some of you may know that a little bit of a rough patch, but thanks to modern technology, I am doing really great. So I've been asked to introduce a section that really has one speaker only. But actually, I will introduce the next few sessions with a few ideas that come from a series of studies. So these are going to be very quick snapshots to tell a story. Okay, I'm going to talk about why I believe we need to rethink our approach to regulation, and maybe some ideas that we need to share based on our studies, both with stakeholders, so people that are in the field, that do this day by day, and also with regulators. And some of these things is pretty cutting edge, so I think we also need to digest it and process it, and then share it with others. And what I'm talking about today is the issue of introduction of pathogens. So introduction of pathogens and regulations that come to deal with such introductions, both internationally but also nationally, because we know that sometimes the pathways are not particularly well understood, so you never know where a microbe may have been before it shows up, especially in a natural ecosystem. And I'm going to talk about how these pathogens are really not what we think they may be based on published literature. And when we think about introductions, we normally can split introduction in two major pathways. And one is a pathway where a pathogen is inadvertently moved from the wild or from a quasi-wild situation in one region of the world and then is moved directly into another region of the world. And of course, in that instance, we're talking about the creation of a new pathosystem. Where there is obviously lack of co-evolution between the host and the pathogen. But then the second pathway is a little bit more insidious, and in the second pathway, a pathogen becomes established in a commodity or in a plant production facility, and then that commodity is responsible for spreading the pathogen both within the region where the commodity is and in between regions because of international trade habits of many industries. So when we think about the first pathway of introduction, and I've brought down some examples to let you know this is not uncommon. Dutch elm disease, chestnut blight, hash dieback, they all kind of fit within the first pathway. There are several factors that are quite important that really should make us think why we need to persist with control of this introduction. Number of genotypes introduced is extremely important. So we shouldn't give up because one pathogen has been introduced, because if we introduce it again and again and again, that pathogen will be much better adapted to invade the new ecosystem. This is something that regulators have a hard time dealing with. And of course, where the genotypes come from is super important. So introduction from different areas brings different abilities, brings different traits, and these different traits together may make for a much better invasion by the organism. So the organism gets different know-hows from different populations. And so avoiding different populations is important. And a third aspect, which is also very important, is of course mating type. And mating type has to do with sex. But we have to think that there's not just sex involved when we introduce both mating types, but there are a lot of genes that are linked to sex genes. And so as a result of this introduction, so when we introduce male and female, we also think about humans. Male and females excel sometimes in different fields. So the same thing applies to pathogens, and it's been proven in many cases. So one more reason to really watch out for these multiple introductions. But today I really want to talk about the second pathway of introduction, and I'm going to talk about the second pathway because it has become a pressing issue in California. In the last issue of 2018 of California Agriculture, we discussed, a bunch of us wrote papers to discuss the issue of multiple Phytophthoras being introduced through restoration efforts. And if you read this nicely written review paper by Bruno Scano and Susan and myself, you will see that at the time we wrote the paper, more than 25 species have been found in these restoration plant production facilities and in restoration themselves. And Laura Simpson is going to talk about that. This is the second pathway. So basically, there is a plant production facility or ornamental plant industry. So there is a commodity that absorbs the pathogen and then becomes responsible for spreading the pathogen both short, medium, and long distance. And my reflections today are going to be on what kind of evidence is there to be concerned about what I call captivity. So I'm talking about when the pathogen is established in one of these commodities, I'm going to say it's in captivity. So it's a period of captivity in a very human artificial ecosystem. And these are the questions that I'm going to ask and answer today with little snapshots. Are there new genotypes that are generated when the pathogen is in captivity, meaning in the plant production facilities? Are novel host by pathogen combinations created in these facilities? Is resistance to fungicides developed? Is there any evidence of increased virulence? And do this facility facilitate hybridization between genetically different groups or genetically different species of pathogens? Generation of new genotypes. I'm going to use very brand new information for most of these questions, either recently published or unpublished yet. This is a study that we finished a couple of years ago, that it's being written right now, about a faint outbreak that we generated at Norsdak, which is the mock nursery in Marin County. And in that facility, we only inoculated 4 genotypes. And those 4 genotypes should be pretty obvious there, those large circles. But 6 and 9 months after, look at those other little circles around. Those are all new genotypes that were generated by themselves inside the facility. And if you think that these genotypes were not fit, look at their frequency. These are the 4 that we inoculated. Look at the frequency of these genotypes as time goes by. So these are not unfit genotypes. These are genotypes that can actually outdo the original genotypes that were already selected because they were extremely fit. Something to think about. So these are not natural genotypes. The genotypes were generated during captivity. And then the work that Marianne Elliott and Takao Kasuga and myself spearheaded about genomic alterations. And this was a work where we had first discovered the process in the nature, in a very harsh environment, which is the oak stem. And then we figured out that nurseries also, in reality, were a harsh environment, possibly because of the use of fungicides. And we found out that the phenotype of isolates in plant production facilities was changing. And what you see here in this picture, and this was due to genomic changes, so changes at the genomic level. And so you have different phenotypes. You have them in the wild, but you also have them in these isolates that are grown in production facilities. Are there novel host by pathogen combination? And here I'm talking about the work of Laura Sims in my lab. She sampled all of the species on the left. She found 9 species, so 9 host species to be infected by exotic Phytophthoras. These were all in California plant production facilities. She found 13 Phytophthora species, and she found 20 new host by pathogen combinations. And actually, we found more, but we reduced that number for political reasons. So yes, the answer, we are creating new pathogenic systems in this production facility. And can resistance to fungicides be developed in captivity? And here we have 2 recent papers. One of my co-authors is right here, Darren Williams, she's going to be talking after me. And we have 2 papers that at the same time were able to prove that in these facilities there is a development of resistance to fungicides. And this was done both with mifenoxam and with phosphites. And what you're seeing here is basically the ability to grow in the presence of fungicides. And what we found out was that we have very close relatives, so very basically brothers, in genetically speaking, and the ones that have gone through the nursery setting, they were resistant to fungicides, and the wild ones were not. And this ability to develop resistance to fungicides means that these isolates are more apt to adapt, so they can adapt much better, because if you can develop resistance to fungicide, you can probably develop other traits rather quickly. Is there evolution of virulence? And here's a paper, again, by Laura Sims, where, and she's going to present this in her talk in more detail, but basically when we look at 2 isolates of the same species, one in nature and one that has gone through a plant production facility, we can actually identify a much higher virulence sometimes in the ones that went through the plant production facility. Granted, this is not a complete sampling, but it's an interesting thing. So what happens in these facilities may actually increase their ability to infect a plant. And are there interspecific or interlineage, which means basically within species mating, but these are lineages that be genetically isolated for a period of time. And without going through all of this verbiage here, the answer is yes. And in our work in California, in these plant production facilities, we found 3 hosts that often were infected by hybrids. And some of these hybrids are recognized hybrids, so they may be already moved around as hybrids, but some of them are new hybrids that we were not really able to define. And interestingly, one of the most common parents, so one of the most common species in these hybrids is Phytophthoras nicotianae, which is, everybody knows, the most common Phytophthora in the nursery setting, without any doubt. So there is a very strong indication that this organization is rampant in these environments. And now I'm going to spend the last couple of minutes talking about, is this really relevant? And I'm going to talk about a very important pathogen, maybe the first pathogen that man has moved around the world effectively. And this is a paper that just came out on Pathology. And the different colors here are different genetically distinct groups. So as you can see, we have 3 genetically distinct groups. And the question was that we were asking, have this group had a different history? Are they associated with different commodities? And are they different in terms of virulence? And what we found out here, those 3 colors here, this doesn't show very well, unfortunately, but there's 3 boxes here, and the WW1, 2, and 3 are the 3 groups. And WW1 is ancestral, so it's the one that includes all the genotypes that we suspected were native or basically ancestral to all the other ones, while WW3 and WW2 developed later on in different parts of the world from WW1. And what you see here are those 2 big circles there. Each circle is a different strain. The bigger the circle, the more frequent it is. And so we have very strong evidence that in this case, in our sample, there were 2 strains that were extremely widespread, extremely ubiquitous almost. And these were relatively recent strains. So these were not ancestral strains, but they derived from ancestral strains. And this is how these strains got to be. So the green one is the original strain, so we're actually able to reconstruct where the pathogen had been before arriving to North America. And in North America, these 2 additional strains, so the purple and the orange strain, got generated. And when we look at their association with different hosts, as you can see, they have a preference for different hosts. So every time there is a release in the wild, of this, that comes from different sources, we have different genotypes being released. And in California, all of these 3 groups have been released in the wild. And our question was, are they different? And this is work that comes from a postdoc in my lab, Maria Serrano, and she was able to prove very clearly that one of these invasive strains, so one of those large circles, one of those derived genotypes, is considerably more aggressive than all of the other strains. And this was a very good representation of the genetics of Phytophthoras cinnamomai. So this is under review right now. So conclusions. Introduced pathogen strains often undergo a period in captivity in human regulated systems. In such artificial environments, they can mutate. They can generate new strains, increase virulence, develop fungicide resistance, hybridize, and increase their ability to adapt, which means they increase their ability to invade natural ecosystems. I believe the regulator should deal differently with subspecies, lineages, and even new genotypes for which we have been able to prove a different phenotype. And also one last thing, which is talking amongst us, we really need to better communicate findings to stakeholders. So there's a lot of research that's been published by people in this room, but I haven't seen any real movement of that knowledge to the people that really need to know this information. And we should blame ourselves for that. And while I'm finishing this presentation, I wanted to tell you there are ways to do a better job. And I'm going to say, because we have tried really hard, and this issue of Phytophthora registration sites, actually, I think we're doing a relatively good job. This issue of California agriculture in December was almost entirely dedicated to Phytophthora restoration efforts. And I worked really hard with UC Berkeley, and if you're at UC Berkeley, unless you win a Nobel Prize, you never get their attention. So I really have to sell my body and soul for UC Berkeley to pick this topic as a topic of interest. But that was an official UC Berkeley press release. So these are just ideas, of course. Citizen science is another one. But we really need to think not just about what our findings be, but also how to better communicate findings to stakeholders, to the public, and to those who need to know the information. Okay, so I thank you. I hope this was interesting to you.